Okay, we'll start again. Welcome everybody, happy Friday. Today we'll talk about the metabolic fates of pyruvate. Pyruvate is the final product of glycolysis, starting from glucose. And when I say metabolic fates, pyruvate can be used for a number of other reactions that go on in the cell. One is to produce lactate. Another is to produce ethanol, but remember this only occurs in yeast. Okay, so we'll talk about that. And pyruvate can also be used to make something called acetyl-CoA. And finally, pyruvate can also be used to eventually make glucose once again in a process known as gluconeogenesis. So we'll talk about gluconeogenesis. I need to have someone track the time for me because sometimes I get carried away in biochemistry and thinking about all these molecules. So I need someone to stop me at about 10:25. So do I have a volunteer that would be willing to interrupt me? Very good. Okay, thank you. So you go ahead and stop me, and then I will return the midterms. We'll go over the key together, and then I want to talk a little bit about this thing called Bloom's taxonomy, which has nothing to do really with biochemistry, but it's a way of learning, and also something called the study cycle, which, to be honest, I wish I had been exposed to these two ideas when I was an undergraduate. So I will expose you guys to these. Okay, so let's get started. And I'm recording this, so hopefully this will be a video when I get it uploaded into Moodle. So last time that I saw you, we had talked about glucose, and how it can be used to create some ATP in the body. And it's going to make two pyruvate molecules, which I'll just abbreviate as PYR, plus two ATPs, plus two NADHs, plus two protons, plus two waters. Okay, so that is the reaction. And it turns out that there are many metabolic fates of pyruvate. One of the metabolic fates is that pyruvate can be reduced to lactic acid, and I'll just show you the reaction here. So there's pyruvate, which, as I said before, you guys should memorize that structure. And then it can react with NADH plus a proton, and will create lactic acid or lactate. Okay. And this is actually occurring whenever we do strenuous exercises. So, for example, if we're running up a flight of stairs and we're using our muscles and we get kind of winded there, this is actually occurring in our muscles. And the reason why it occurs in our muscles is that we need to regenerate this NAD plus in order to be used for glycolysis again. So we have this sort of interesting cycle, which I will show you. And it's a way to quickly regenerate ATP from glycolysis in our muscles. So here we go with sort of a diagram here, which I show how the NADH gets recycled to be used for glycolysis. So here's glucose. And then we have reactions 1 through 5 of glycolysis, and that makes 2 of these glyceraldehyde phosphate. And then we have this enzyme known as glyceraldehyde 3-phosphate dehydrogenase. I'll just abbreviate that as GLY 3-phosphate dehydrogenase. But I'll put these exclamation points around it so you guys know that's an enzyme. So there's a little enzyme there. And that converts 2-glyceraldehyde 3-phosphate into 2 molecules of 1,3 bisphosphoglycerate. Okay. And then we have reactions 7 through 9 of glycolysis. And then down here we're going to make 2 molecules of phosphoenolpyruvate and then in reaction 10, so here's reaction 10, we're going to make 2 molecules of pyruvate. And then, as I said, in strenuous exercise conditions, the pyruvate can go on to make 2 lactates. And the enzyme that does this is called lactate dehydrogenase. Sorry. Lactate dehydrogenase. I put these exclamation points around so you know that's an enzyme. Okay. And what cycles between this, between these two, is you have 2 molecules of NAD plus, and that's necessary for this reaction. Okay, so that's necessary for reaction number 6 in glycolysis, glyceraldehyde 3-phosphate dehydrogenase. And we're going to make 2 molecules of NADH plus 2 protons. Okay. And then this actually goes here, and those will react with lactate dehydrogenase and pyruvate in order to create your NAD+. Okay, so that's how that cycle kind of goes. And why do we need to do this? Well, in order for glycolysis to keep on going, you have to have a steady supply of NAD+. And NAD+ is limiting inside the cell. So the cell doesn't have very much of this NAD+. It just doesn't retain that much of a pool of NAD+. So we got to regenerate this. If we didn't have this ability, then we wouldn't be able to perform a vigorous exercise, and we would be in trouble. The other thing that this does is that actually the lactate is acidic. Okay, this is acidic, and it gets released from the muscles. It gets released from the muscles into the bloodstream. So the lactate gets released from the muscles and it goes into the bloodstream. Okay. And this causes the, because lactic acid actually is an acid, okay, this causes the pH of the blood to go down. Okay, pH decreases in blood. Okay, and I don't know if you guys went over this in Chem 431A, but does anybody remember what a lowering of the pH does to hemoglobin? What does that do to hemoglobin? Did you guys go over hemoglobin at all? Okay, good, good. Excuse me? Higher binding affinity. Very good. Lower binding affinity. Okay, so this actually will lead to a decrease in the oxygen binding affinity of hemoglobin, and this causes our hemoglobin to more efficiently deliver the oxygen to our tissues, okay? And then that hemoglobin goes up into our lungs and can capture more oxygen and deliver it more efficiently. So I'm going to say an increase in delivery of O2 to tissues, okay? Because now the hemoglobin doesn't bind so tightly to the oxygen, and that oxygen can be released to the tissues more efficiently. And this basically causes us to want to grab more oxygen, and so we get out of breath, and so we want to, that's why we kind of gasp a little bit. People like me, I gasp, you know, after running 100 yards. And so that's because I'm trying to breathe in more oxygen because of this effect. So it all kind of fits together with a hemoglobin and its affinity for oxygen. Okay. Eventually, also, you'll notice that your muscles kind of ache a little bit, right? And that's caused by the acidity also, the acidity in the blood, and the acidity actually near your tissues. It causes that sort of pain in your legs and your arms as you make those last flight of stairs up from the bus station onto the campus. It's tough. Okay, so that's how it works. So that's an interesting phenomenon, and we'll return. Oh, by the way, eventually, when you catch your breath and you more efficiently get rid of pyruvate, there's another way of using pyruvate, and the other way of using pyruvate in the humans is to make something called acetyl-CoA. And we'll talk about that in just a second. So there's another metabolic fate of breakdown of pyruvate, and that's called the production of acetyl-CoA and CO2. By the way, if you look very carefully at pyruvate and compare it to glucose, you will find that on a per carbon basis, glucose is more reduced than pyruvate. In other words, pyruvate is more oxidized. And eventually, all the carbons that originally came from glucose will be oxidized fully to CO2. So that's eventually going to happen. But at this point, you should convince yourself that pyruvate is more oxidized than glucose. Okay. I'm going to switch now to an important industry, an important phenomenon, which we find in yeast. Okay, so other organisms have a slightly different way of recreating the NADH to be used for glycolysis. So we use lactic acid, okay, but yeast, they do something a little bit different, okay? So we're now going to talk about yeast, and we're going to talk about pyruvate metabolism in yeast. Okay. So in yeast, we have again, I'll draw pyruvate. Okay. And then we have an enzyme known as pyruvate decarboxylase. Okay. And what this will do is this will react with NAD, I'm sorry, it doesn't react with anything like that. Okay, pyruvate decarboxylase. And what we're going to make here is CO2 plus this compound and that compound, that molecule is known as acetaldehyde. Acetaldehyde. Okay. I'm going to go through this exercise with you so that we're all on the same page. I'm going to show you that overall there's been no oxidation or reduction done here. And we'll use these oxidation numbers for this little task here. Okay? So let's work together with me. This is CH3. Okay? And so can someone tell me, can someone tell me what is the oxidation number of that carbon right there? -3. Very good. Okay. And how about this central carbon right here? Come on, be brave. Shout it out. +2. Okay. All right. +2. Okay. And what about that carbon? +3. It has 3 bonds to oxygen, right? So that's going to be +3. Okay. So if we add up all of these, we're going to get a +2 for this guy. And what I'm also going to do is I'm going to divide it by the number of carbons. So I have 3 carbons down here in this molecule. So my oxidation state is going to be +0.67, right? So I just took 2/3. Okay. Now, let's go ahead and do these guys. CO2. What is the oxidation state of carbon there? +4. Excellent. +4. Okay. What about this carbon here, acetaldehyde, this aldehyde carbon? +1. Very good, very good. So you took the +2 from the carbon bound to oxygen and you subtracted the 1 for the hydrogen. Very good, that's +1. And then this guy, we already know that, we did that before, that's going to be a -3. Okay, that's the methyl group. So if I add up all of these numbers, let's go ahead and add up all of these numbers. I'm going to get +2, right? We get 5 minus 3, okay, it's going to be +2, okay? And the total number of carbons here, even between these two, is going to be 3 carbons, okay? I'll just say 3 carbons. And that's going to be also a +0.67. So this enzyme actually belongs to the lyase class. Okay. All right. Not an oxidoreductase here. Okay. Then the next step that the yeast does, so the yeast does this in 2 steps. It's going to take acetaldehyde, and we have this alcohol dehydrogenase. So that's the name of this enzyme, alcohol dehydrogenase. And we've got NADH plus a proton, and that's going to get converted into NAD plus. And can anyone name the product that I'm gonna write down here? Someone said it. Ethanol. Very good. Ethanol. So we're going to get this, okay? So that is ethanol. And so yeast has a couple of useful purposes for humans, right? One is that you can go to pavilions or Ralph's and you pick up these little packets, especially around Thanksgiving time, and these little packets say yeast activated, right? Little packets. And if you work with dough, you can sprinkle that into the dough, and you can grease up a little bread pan, and they tell you to put this kind of in a high place that's warm. I usually put it above the refrigerator, and I usually cover it to keep it kind of warm. Try to keep it near 30 or 37, about 37 degrees if possible. And what happens is that bread rises, right? And the reason why it rises is that it's actually putting off this CO2 gas. That CO2 gas is what's causing that bread to rise, okay? Of course, the yeast really needs it for this step right here. So this is the second step that it does, because this is necessary for what metabolic pathway in order to keep on going? What is that NAD plus necessary for yeast? What pathway is NAD plus necessary? Anybody know? What catabolic metabolic pathway is it necessary for? What catabolic necessary? What would you need NAD plus for in this area here? Can anyone name the pathway? Glycolysis. Yes, glycolysis. Yes, I know you guys are thinking like a trick answer. Oh, something that we haven't studied yet. No. It is glycolysis. So basically, this functioning yeast is doing the same thing here, except it's not making two lactates. It's making two ethanol molecules. Okay. And then, of course, the next step that the yeast does is it also makes ethanol. So our bread actually does have a little bit of ethanol in it, uncooked. But when we put it in the oven, that ethanol evaporates, and so that's why you don't get drunk eating bread. Okay. But of course, there's another industry that uses this ethanol producing capability, and that's the wine industry, right? So wines obviously are made from yeast producing ethanol, and eventually that ethanol gets to be at such high levels that it starts to kill off the yeast. Actually, that's what happens. And it's like they're drowning in their own waste product there, the ethanol. So the ethanol isn't used by the yeast. It's actually a product because they're always trying to make this NAD plus. Okay, so you feed some yeast some sugar, and eventually they're going to make some ethanol. Okay. Next, I want to talk about another, actually before we go on to that, I'm going to talk about these things called radio labeling experiments. Radio labeling experiments. Okay, so I had told you at the end of last lecture, near the end of last lecture, that there is this way that organic chemists would radio label the glucose so that they could trace where the different carbons were coming from in the products. And I had told you that if we were to write down the structure of glucose here, that's the structure of glucose, that you can actually draw a line through halfway through the glucose. Try and make that a dotted line, okay? So that you have two halves of the glucose, and eventually these will become two molecules of pyruvate. drawing these two molecules of pyruvate. And if you follow very carefully how the enzymes, how the series of enzymes make these two molecules of pyruvate, you will find that this carbon right here makes that carboxylic acid, and this carbon right here makes that carboxylic acid. And then of course, this second carbon in pyruvate is going to be this carbon right here, and the third carbon in the pyruvate is going to be this carbon right here. That's for this molecule. And then the second carbon in pyruvate will be this one right here, and the final carbon here will be this one right here. Okay, so we got how these are made. And let's go ahead and do a couple of interesting problems. Let's pretend, let's say this is a question here. The question is that glucose is labeled at carbon number 1. Okay. Give how many, actually, give the, let me just redo this. Okay. Give the percentage of pyruvate molecules that will be labeled and the location in pyruvate where the label will be. Okay? And the label that we always, I shouldn't say always, but we oftentimes will use is C14. So C14 is the radioactive label that we use. Okay, so let's see if we can't do this together. Okay, so we're saying that glucose is labeled on the number 1 carbons. Well, it helps if we know our numbering system. So review your numbering system. Okay, the number 1 carbon for glucose is right there. So that's going to be labeled. I'll put a little asterisk right there. That's the label. Okay, and I know that out of 100 molecules, let's say I had 100 molecules of glucose, they're going to make 100 molecules of pyruvate. Actually, it's going to be 200 molecules of pyruvate, but I'm going to break it up into two 100 sections. So here's 100 molecules of pyruvate. Here's another 100 molecules of pyruvate. Because you know that for every glucose, you make 2 pyruvates. Okay? And so the question that I'm going to pose to you is, which numbered carbon in pyruvate is radiolabeled? So in pyruvate has 3 carbons. Which numbered carbon in pyruvate is going to be radiolabeled? The methyl group 1, perfect. Can we give a number to it? It's either got to be 1, 2, or 3. 3. Okay, and you knew it was 3 because the way that we label, we number these is that this gets the highest number, this gets the second highest number, this gets the third highest. So this requires you to know your numbering system as well, right? Very good. So that's exactly right. The methyl group, or the number 3 carbon, will be labeled in pyruvate. And what percentage of the pyruvates will be radiolabeled? Beautiful, 50%, exactly. So you notice that I split this up into two. So basically, only 50% of the, only 100 out of the 200 pyruvates made will have the radiolabel, okay? Now let's say you're working for a company that makes these compounds, and your customer wants to have all of the pyruvates radiolabeled in the number 3 position, okay? And you're starting off with glucose because your organic chemist only knows how to make glucose, okay? So my question to you is, how can we make a glucose so that we make sure that 100% of the pyruvates are radiolabeled at the number 3 position? Where do we need to put the radiolabel in the glucose? Not 3 and 6. It's got to be 1. We know 1 for sure, right? And the other one is going to be in number 6. Very good. Okay, it's got to be right here. If you were able to feed your bacteria or feed whatever it is, whatever organism you have, this, then you know that at the end of the day you're going to get radiolabel in all the pyruvates, and all the pyruvates will have it right here. Okay, very good. The other thing that you could do is you could start off with glyceraldehyde 3-phosphate. Okay, great. So I would suggest that you guys do problem number 5 for, I don't have time to go over that. We will now go ahead and talk about the exam. Before we do that, and this is related to that, I want to go over this idea about how to study. I know actually surveys were made of students in just beginning in college about what it would take to get an A in a high school class. And basically, the students responded that all they needed to do was remember the material and basically regurgitate that for the test. You're going to get an A. You're going to get an A in high school. And they've done several surveys, thousands of surveys showing this. But when they asked the same college students in the same survey in terms of what it would take to get an A in a college class, and they haven't had that many college classes. We're talking about freshmen here. They said basically, doing something creative with that knowledge, okay? Putting it together in a new form and being able to teach it to others. That's the kind of knowledge that you have to have in order to do well in college. And so this is a pretty old Bloom's taxonomy, but it really talks about a pyramid of learning here. The lowest form is just remembering. The highest form is being able to use your knowledge to create new patterns and new ideas, okay? And what we hope that to do is that by the time you guys graduate, that you're basically way up here and you're not just remembering, because that's just not going to be enough in college classes, okay? I also want to mention that there's something called the study cycle, and in the study cycle, it requires not very much time, but really focused effort. And this is actually part of a lecture that was given to us by a professor at LSU, Louisiana State University. And it just goes through this sort of pyramid here, or maybe these concentric circles. Preview before you come to class what things we're going to be discussing in the chapter. obviously attend class, okay, and then answer and ask meaningful questions and take good notes, okay. Review right after class if possible, as soon as possible. I think I said that in the very first day, okay. And read the notes and fill in the gaps. So anything that you didn't understand, go to the book and fill in those gaps. And then obviously study, okay, so study. Sorry, this is so small for you. Study, okay. And do intense study sessions and weekend review, and then assess your learning. Really figure out if you're really learning. Are you really understanding the material enough so that you can go to your dinner table and basically teach your significant other, or you go to your family and you can actually talk about what concepts you've learned, and you can teach it to them, okay? And this sort of gives you the number of minutes to do this sort of intense studying that you'll need. This doesn't include, doing the problems, which is also part of it, and doing the sapling homework, but I think this might be a good way to review this. And I've never actually talked about this before this class. I've never incorporated this material, but I was very inspired by the person who spoke. Her name is Sandra McGuire. She even has a book. I even bought the book off of her website in order to help students be able to do well in biochemistry. At the end of the day, you're competing with lots of folks that are biochemistry majors, and folks from UCLA, UC Berkeley. My daughter, she went to UC Berkeley, and I used to visit her. She wasn't a biochemistry major, but she had roommates that were biochemistry majors. And I would talk to them and ask them, well, what are the kinds of things that you guys are learning? It's pretty intense, pretty intense. What are your tests like? Very intense. And they would show me some of the problems, and they are similar to the problems that I've given you on your first midterm here. So I'm making a case that the rigor that we're working at, the mechanisms, the problems, the radiolabeling experiments, that's the kind of stuff that your competition is also being exposed to. People from UCLA, UC Berkeley. And so in order to get those jobs out there, you need to perform well on standardized tests and interviews so that you know your stuff. So I'm making a case for the kind of rigorous material that I'm giving you. Okay, that said, the high on the exam was 86 out of 100, and the average was about 57, 58. So average is a little bit low. Okay, so I'm going to hand these to you. Oh, here they are. But I need you to look at them, make sure that I tallied up the points correctly, just like your quiz, and then I need them back. You can always come to me during my office hours and spend as much time as you need looking at your test. But right now, look at it. If we have a chance, we'll go through some of the questions together, and but I will need the test back. And if you guys could turn off your recorders, I would appreciate it. Okay, let's go ahead and hand these back. Christina. Maria. Joanna. Philip. Just do something here.